Now, if we look at SFC, honestly, the screening approach is very similar to normal phase method development. It's the same 12-column switching valve. It's a little bit different, obviously, in that you've got a modifier and CO2 as your primary mobile phase components. The screening set basically looks the same as well. It's covering all of the immobilized columns and some of the coated phases. The mobile phase combinations, as I mentioned before, obviously going to be different. This is SFC, so supercritical CO2 is your primary mobile phase component. It's essentially the hexane of your normal phase separations. So we'll be screening for the coated phases, CO2 alcohols including methanol, ethanol, and IPA. And we can also look at acetonitrile as well as a modifier. For immobilized phases, normal phase again is the same, but we now have access to the extended range solvents, addition of DCM, THF, or MTBE. Because those solvents oftentimes don't have the greatest miscibility with supercritical CO2, and they can cause some issues with phase separation. 20% or 10% methanol is oftentimes helpful, not only in helping with that phase separation, but also just in getting things to come off the column a little bit faster. DCM and THF and MTBE are not the most polar solvents, and so sometimes things can simply be retained for a little too long. Not surprisingly, the modifier has a dramatic effect on the peak shape. It also has a dramatic effect on the elution order, as it did in normal phase separations. Sometimes you can see a reversal of elution order. In the case of methanol and IPA, it has been observed dramatically where, like I said, it can actually reverse the elution order. A few examples of the modifier effects here. On a sample compound, this benzoin ethyl ether, changing the modifier, just like changing the mobile phase component in normal phase, can elicit some pretty dramatic differences in the separation, assuming you are keeping the CSP, the chiral stationary phase, the same. Just a few more examples here of screening for methocarbamol on IA, IBN, and IC. Here, that kind of demonstrating that even by changing the chiral stationary phase, changing the mobile phase, you can get nice baseline resolutions. It's all not predictable. I can assure you that the screening approach is extremely helpful in these cases. And then one more example here for Devronol, the same thing. New separations or interesting separations as a result of the screening process. Optimization looks exactly the same as it does for normal phase. We can look at changing the temperature. We can look at changing the column length. We can look at changing the flow rate. This is kind of an all-inclusive example here on CHIRALPAK ADH. We go to a longer column. We go to a slower flow rate, and we decrease the amount of modifier, and we obviously have an improvement in the retention from less than a minute to just a little less than 5 minutes, and a nice baseline resolution as a result of the optimization process here. Mobile phase additives look very similar as well, except for acidic additives. Oftentimes we don't need to add any acidic additive to SFC separations. Supercritical fluid, supercritical CO2 does have some acidic characteristics to it. The carbonic acid that is present is often enough to help improve the peak shape of the acidic compounds. But if you do find a particularly acidic compound, you can add acetic acid or formic acid or TFA. Amines is basically the same, DEA, TEA. You can use some of these linear or cyclic amines like cyclohexylamine. And for amino acids or amphoteric molecules, again, you'll be looking at TFA and purifying or separating the ionized or the ammonium salt of the free base. Similarly, since we're essentially using the same columns, it's 0.1% usually is enough. For bases, because there is some acidic characteristics to the supercritical CO2, you might need 0.2% to kind of help neutralize that carbonic acid a little bit. But again, not exceeding 0.5% to help improve the lifetime of the columns. A resource for you. This was published a few years back, particularly exploring the use of those extended range solvents. This was done in collaboration with Pfizer up in Groton, Connecticut. Some interesting strategies for screening on the immobilized CHIRALPAK line. We did not have IG through IJ at that point, so this is only including IA through IF. All right, we still have a few minutes left here, so I will briefly touch on those new products that I mentioned. We've got sub 2 micron columns, which are optimized for UHPLC use. They can be used on a normal HPLC, however, to get the full benefit of the reduced extra column band broadening and the reduced or the increase in the plate count, UHPLC is certainly the way to go. IA, IB, ID, IC, IG, and IH are the 6 columns that we have. Very good for multi-component analysis here. This is an example with cannabinoids on CHIRALPAK IBU. Normally under even 3 micron conditions, you see some of these really tight separations here. These would probably end up co-eluting or potentially, baseline, 50% baseline, something along those lines. But because you can push these columns a lot faster, you can obviously decrease the flow rate. You can decrease the analysis time as well, getting some really ultra fast separations, really help you power through those intense screening sets that you might have if you've got a micro well plate or something like that. A significant time savings here by switching to a short column, switching to the small particle size, allowing you to go ultra fast. And I also mentioned at the beginning too, our newest phase, CHIRALPAK IJ, which is the immobilized version of ChiralCell OJ. It's cellulose derivatized with tris 4-methylphenylbenzoate. Very similar separations to ChiralCell OJ. So if you're looking to transition over to a more robust column that can withstand some of those extended range solvents, IJ is definitely a great option. Here's 3 examples of that. And in some cases, we do have some new or improved separations with the access to those extended range solvents. 2 examples here, 5-methyl-5-phenylhetantane, a nice separation here with hexane THF. And for, I won't try to say the name and botch it here, but no separation actually, or partial separation using the OJ on normal phase, you now have a nice baseline resolution under extended range conditions. So nice new addition to the screening set from the older OJ version. To finish up today, I'll just highlight a few things on our website. Over here on the left-hand side is the address you can visit us at. On the right-hand side here are some quick links, a lot of really good things to look at. Here's the method development strategies link that I mentioned before, but we'll look at the instruction manuals. All of our instruction manuals are available electronically now online. I highly recommend that you look at those. They have some method development strategies in there, some recommended starting conditions, as well as suggestions for additives, regeneration procedures, and so forth. We're currently in the process of overhauling all of these instruction manuals. So if you have versions of these, I would encourage you to check back in the next few months to get the updated versions. They'll have IJ in there, obviously, as well as some new tips perhaps that were not in the old ones. And then here is the email address, since we are basically out of time, that you can email if you have questions about this presentation, or if you just have general chromatography questions, please feel free to email us at questions@cti.daicel.com. Feel free to email me as well if you have questions about the presentation. We have a number of other webinars that we've done in the past, things on chiral method development, sub 2 micron, chiral stationary phases, method validation, and so forth. And we do have one more webinar scheduled for the end of this year The topic at the moment is to be determined. So if you have some suggestions on things that you'd like to see, I would encourage you to email me. I'd be more than happy to try to incorporate that and make a webinar that's perhaps suited to something that is very interesting to you. So at this time, I will pass it back over to Tracy to wrap things up for the day, but I appreciate you joining us once again, and here she is.
Thanks, Wes, and thanks to all of you for attending. We hope you enjoyed our webinar. Please do not hesitate to contact us with any questions and stay tuned for our lineup of webinars coming later in the year. Have a great day. Bye-bye.